you that how to use this particular, this particular Python code which I had given you, how to use this code to import sequence from NCBI. Okay, so for that, what you will have to do is that you will have to open your Google Collaboratory. Okay, or if you are using Jupyter Notebook, you can open that, or if you are using Visual Studio Code or PyCharm, you can open it up. Here I'm gonna use Google Collaboratory. Okay, so I've opened the new notebook in the Google Collab. First of all, what you will have to do is that you will have to write down pip install biopython. Okay, and after that I'm going to run this code, and it's going to take some time, and biopython will be installed in our system. Okay, requirement already satisfied, that means we already have biopython. And now what we will have to do on here is that we will have to click on plus code, and then what you simply have to do is that you just have to copy this code. Okay, just have to copy this code. I'm gonna copy it, and you just have to paste it on here. Okay, and inside of this code, I'm gonna import Entrez. I'm gonna explain how this code actually works. From bio import Entrez. So what is Entrez? Entrez is actually a molecular biology. It's already written on here what is Entrez, but I'm gonna explain to you that Entrez is basically a molecular biology database system, and it provides integrated access to nucleotide and protein sequence data that is provided by NCBI website. Okay, so from bio we are going to import Entrez and sequence input output, and here Entrez.email, it asks for your email. So here you can write down your email. Here I'm going to write down my email. Whatever is my email, I'm going to write down it. You will have to write down your own email. Okay, and after that this code will go like that. Okay, and after just writing down your email, what main important thing is on here is that inside of this handle. Okay, inside of this handle, first of all, Entrez.esearch, internet search database. So as you told me that you're going to search for the nucleotide, so write down nucleotide on here, and uh, then inside of this term you will have to write down the gene name. Okay, gene name. So suppose if you're gonna search for the mRNA or any kind of DNA, then you will have to specify the gene name, and inside of this gene name either you can write down the accession number, gene ID, or directly the name of the gene. So here I'm directly gonna write down the name of the gene, because here I'm gonna show you the example of CRT gene, that is called as chloroquine resistance transporter. Okay, this CRT gene is found in Plasmodium falciparum. Plasmodium falciparum is a bacteria which causes deadliest form of malaria in human beings. Okay, so I'm gonna search for CRT gene, that is chloroquine resistance transporter. I have just written the name of the gene. Okay, uh, it's also gonna work. Or in place of the CRT, you can also write down the accession number or the gene bank ID. Okay, but better if you write down the name. Okay, and after that, nomenclature of the organism. So as I told you that I'm gonna work on the CRT gene of Plasmodium falciparum, which causes the deadliest form of malaria. So here nomenclature of organism mean the scientific name of organism. So here I'm going to write down Plasmodium falciparum. Falciparum. And always make sure that the spelling of the organism is correct. Okay, falciparum. And after that, as you can see, a retmax is 40. So by default, retmax is 20. Here I'm going to write down retmax 40. So what does it mean? It means that it's going to give you, as an outcome, it's going to give you 40 records back. You can make it 1000, 2000, whatever is in your mind, whatever sequence, how many amounts of sequence you want. You can fix it like 1000, 2000. But here by default it's going to be 20, but here I'm going to make it 40, because as much number you will as bigger number you will add, it's gonna create a kind of load on the server. So here, to just explain it, I'm gonna keep it 40. You can fix it as much as you want, like 1 million, 2 million, anything, but make sure that it shouldn't put any kind of load on the server because so many people are using it. Okay, so up to this, you will have to write down your email, and inside of this handle, just to write down inside of this term either the name of the gene, accession number, or gene bank ID. Anything better if you write down the name of the gene. Here the name of the gene in Plasmodium falciparum is CRT, that is chloroquine resistant transporter. Okay, and after that, just write down these codes. These are just the codes you will have to write down, length and everything, and it's gonna print out at the end. It's gonna print out the sequence and other details. Okay, okay, and now what you will have to do is that you will have to click on this play button to run the code. So it's going to take some time. It's going to take some time, so we will wait for that. Okay, so code run is successful, and it says Plasmodium falciparum isolate 22013707 for chloroquine resistant transfer gene, and it has given you these sequences. Okay, as I had told you that retmax is gonna be 40. Retmax is gonna be 40. So it's gonna give you 40 records on here. You can fix it according to your need. You can make it 1000, 2000, whatever sequence you want. But these are the sequence. Okay. So I hope this point is clear right now. So basically, what you will have to do is that this is the code. You can't, like this is the code. You just have to copy and paste it on here, and inside of this code, you just have to do is that you just have to write down your email is necessary to write down on here, and after that, inside of this handle, you just have to write down the name of the gene inside of this term, or either you can also write down the accession number. Okay, no problem. I remember the gene name, or better if you write the gene name. Okay, and the name of the organism. Make sure that the name should be in the form of nomenclature, scientific name, spelling should be correct, and running this code is going to give you the result. Okay, I hope this point is clear right now. Thank you.